So today I'm presenting a prokaryotic replication fork with random household items, and I'll explain and elaborate each aspect of the DNA replication fork, starting with the origin of replication. And it's not actually seen in this diagram because this is just half of a replication bubble, and the origin is usually right in the middle. So there's usually only one origin of replication in a prokaryote, and it is the location at which replication begins, and it's characterized by a unique sequence of nucleotides, which are then recognized by an initiator protein, and once the origin is recognized, a protein called helicase, which here is represented by pistachio shell, can come in to unwind the DNA double helix, and it does this by breaking the hydrogen bonds between the base pairs of the two strands. And in order to maintain a stable and functional replication fork, we need single-stranded binding proteins, which are the little yellow circles, and we need gyrase, which is the scrunchie at the top. Single-stranded binding proteins bind to the single strands of DNA to protect them and ensure that they stay separated and don't bind back together, and to prevent hairpins. And gyrase is important because it reduces torsional stress, because as we're unwinding the double helix, it can cause supercoils and a lot of torsional stress, and the gyrase relaxes that so we can go along with replication. And in order to do that, we need a free 3' hydroxyl end, which DNA can't actually supply because its free 3 end is just hydrogen. So RNA primase comes in to supply the free 3' end by producing RNA primers. And the primase is represented by the cashew, and the primers are represented by the blue parts of the pipe cleaner on the left side. And the next important structure is DNA polymerase 3, which is what attaches to the RNA primers to add nucleotides to the free 3' hydroxyl end on the primers. And that is represented by the green puffs on either side. And since replication occurs in the 5' to 3' direction and the two DNA strands are antiparallel, one strand of new DNA will be synthesized continuously, and this is called the leading strand, and it's represented by the purple pipe cleaner. And on the other strand is discontinuous DNA synthesis, and this is called the lagging strand, represented by all the yellow pipe cleaners. And since it's discontinuous, instead of one long strand, there's many short chunks called Okazaki fragments. And in order to get rid of the RNA primers at the end of each Okazaki fragment, we need DNA polymerase 1, which is the little basketball sticker. And this is going to break down the RNA primer and replace it with DNA nucleotides so that our strand is composed exclusively of DNA and no more RNA. And since we're still cut up into fragments on the lagging strand side, we need ligase to come in and seal the new strand. And it does this by forming a phosphodiester bond between the nucleotides by linking the 3' hydroxyl end of one nucleotide to the 5' phosphate end of another. And that's the prokaryote replication fork.